I'm going to speak about the history of crystallization, which does in fact go back almost 200 years, not quite 200 years. But before I begin, I should say that a much more thorough treatment of the history of crystallization has appeared recently, and this was written by Richard Giget, and it was in Feb's Journal, volume 280, 2013. So if you want a really good, thorough treatment of everything that's happened in the past 200 years, then I think that's the place to look. Now, as I go through this, I'm going to talk principally about crystallization, but you're going to see at a certain point that the history of crystallization of macromolecules becomes inseparable from really the history of X-ray crystallography of macromolecules. And so those two things are going to become intertwined, and it's going to be very difficult to separate those two. I'm not going to try to separate those two. So let me go back to the initial event, and that was in 1840. A German scientist by the name of Hunefeld was the first, so far as we know, was the first to crystallize a protein of any sort, and the protein that he crystallized was hemoglobin. So hemoglobin has a first not only as being one of the first structures determined, but also being the first protein that was crystallized as well. And he crystallized this protein simply by taking a bloodworm and squeezing its blood out onto a microscope slide. And as that blood evaporated, he noticed that these red crystals formed, and that these were in fact the hemoglobin crystals. And this was then repeated by a number of other, always German scientists, for about the next 20 years in laboratories, in which they squeezed or somehow got the blood out of a whole series of different organisms, mostly animals, of course. And they noted also that these hemoglobins crystallized by simple evaporation. So this really was the first demonstration that dehydration of proteins will produce some crystalline material, at least in good cases. Now, this was basically a laboratory curiosity for the next 20 or 30 or even 40 years, because there was no realization of the impact or what this really was telling people. Proteins were not understood at that time. The first amino acid, asparagine, had only been recently discovered at the time of this event. So people didn't even know that proteins were polymers and made up of amino acids, so that wasn't appreciated. And in addition to that, of course, at this time, crystals really, the physical nature of crystals wasn't understood as well. So the real meaning of this was at this time lost. Now, in the 1880s, there was an agricultural chemist in Connecticut who's long forgotten now for the most part. His name was T.B. Osborne. And Osborne realized, of course, that crystallization could be used as a purification tool. And he was attempting to crystallize the proteins from a number of different nuts and plant seeds. And he used crystallization as a technique to do this. And he crystallized something like a half a dozen different plant seed proteins. One of them from Brazil nut. This was, I think, a major success of his. He grew these very large crystals of Brazil nut protein, but a number of other proteins as well, principally from plant seeds. And the interesting thing about his work is if you go back and look at his papers from the 1880s, and indeed I did this, they were in the library at MIT where I was a postdoc once. I went back and looked at these papers, and he was the one really who first introduced the idea of using salt fractionation to separate different proteins. He was the first one to use temperature to help separate proteins. He was the first one to use alcohols in organic solvents of various sorts in order to separate and crystallize proteins. So he didn't really realize where this was all going, but he was using this strictly as a purification tool. And he didn't even consider himself really a crystallographer. He was simply a plant biochemist. But he introduced many of these techniques now that are still in use by us today. In 1898, the second animal protein was crystallized, and this was by a pair of physicians, Hopkins and Pincus, and they crystallized egg albumin. And shortly after this, Hofmeister, who everyone is familiar with, reproduced their work and also carried out additional work on egg albumin and the crystallization properties of ovalbumin. And the interesting thing about their work is, if you look at their papers, they were the first to realize that you could use pH as a tool to crystallize proteins. So in fact, they used variation of pH, variation of ammonium sulfate concentration, and variation of temperature in order to crystallize proteins. And these, of course, are all things that we do today in the laboratory still to crystallize proteins. Now, the real giant of protein crystallization was J.B. Sumner. J.B. Sumner was, again, an agricultural biochemist, and he was at Cornell University at the Agricultural Experiment Station in Ithaca, New York. And Sumner had this belief that enzymes were proteins, and that proteins, these enzyme proteins, had discrete three-dimensional structures. Now you might say, well, isn't that obvious to us? Didn't we always know that? And of course that wasn't true. For a very, very long time, that was a very, very controversial idea, because many, many prominent scientists believed, in fact, that proteins were a sort of a colloid, that it was aggregates of proteins that, in fact, carried out the actual catalysis, and that you could not ascribe to a single protein molecule the catalytic function that was known to exist. But Sumner didn't believe that. Sumner believed what we know today to be true. And he set out in early days, in the early 1900s, to crystallize an enzyme, to actually crystallize a protein molecule which he could then demonstrate had an enzymatic activity. And he set about to crystallize, to purify and crystallize the enzyme urease from the jack bean. The jack bean is a lowly bean that's a lot like our lima bean. It's used as a cattle feed, really. But it was known that the enzyme urease was present in the jack bean. So he set about trying to crystallize urease from jack bean. Now, he was not initially successful. In fact, for many, many years, he was unable to do this. But along the way, he, by fortuitous chance, crystallized a couple or three other proteins as well. One of these is concanavalin A, and one of these is concanavalin B, which he crystallized and published in 1919. Now, we know now that concanavalin A is the lectin. It was the first lectin that we've studied. It's a very well-known molecule because it's used in cell biology work as well. So it was the first lectin, but of course he didn't know it was a lectin. He didn't know what it did. He simply called it concanavalin A after the plant seed. The other protein, interestingly, concanavalin B, was subsequently determined to be the enzyme chitinase. And again, he didn't know that function, but in fact, he had already achieved his purpose with this 1919 work. He had, in fact, crystallized the first enzyme. He had crystallized the enzyme chitinase, but he didn't know that. So he continued on to try to crystallize this enzyme called urease. And he succeeded in 1926. And by crystallizing urease, and in fact showing that crystalline protein, which by then, of course, implied that it did have a discrete three-dimensional structure, because we now knew what crystals were about, the physical properties of crystals, he was able to demonstrate that he had crystallized something with a discrete, unique three-dimensional structure, and that had urease activity. This was questioned for a number of years. People thought that perhaps he had co-crystallized some impurities, which in fact carried the function, but that was shown not to be true. Now, as Sumner was doing this work, and he was plodding along here more or less in obscurity until he was able to carry this out, other things were happening, and we know those, of course, from other historical accounts. We had Sir William Bragg, who was the first to build the X-ray spectrometer and first to apply that to crystals, and his son, Sir Lawrence Bragg, both of whom were awarded the Nobel Prize, a pair of Australians, father and son, who were awarded the Nobel Prize for their early work, not on proteins, but they were developing the tools, they were developing the techniques for the study of ultimately macromolecular crystals. And we had a very important event occur in about 1927. And this was by a team. The first author was Abel, and they worked for Novo Nordisk company, the Danish pharmaceutical company, in fact, in Copenhagen. And what they accomplished was the crystallization of the protein insulin. Some people may argue, well, insulin's not a protein, it's just a little peptide. No, it wasn't. It was the first protein, really. Or it was a protein. And they crystallized insulin, and this was extremely important. You might say, well, okay, other things have been crystallized, hemoglobin and ovalbumin and all of these other proteins now, the ones that Sumner had done. What was so important about this? And the importance of this is that in the early 20th century, insulin was one of the most important drugs that we knew about, because until that time, we had no way of treating diabetes except with extracts of pancreas of hogs and cows. And these extracts, of course, were heavily contaminated with other proteins, They induced terrible immune responses in patients, and ultimately those extracts were of really very, very little prolonged value. But by crystallizing insulin and getting it in this very pure form, he was able then to overcome that immunological response. And in fact, that's what really kept diabetics alive, I think, for the rest of the century. And of course, we're still crystallizing proteins today. The important feature of Gall's work, so far as our understanding of how to crystallize proteins goes, is that he was the first one to use metal ions, as well as pH, as well as salts, as well as temperature. So he introduced the idea of using various ions, in this case cations, in order to crystallize proteins. Now, another major figure in crystallization of the 20th century was Northrop at Rockefeller University in New York. And Northrop's group, which included a technician by the name of Moses Kunitz, who ultimately became almost as well known as Northrop. Moses Kunitz and Northrop set about crystallizing proteins from the pancreas, and they were tremendously successful in this. They crystallized ribonuclease, they crystallized trypsin, they crystallized trypsinogen, chymotrypsinogen, pepsin, a whole series of different pancreatic enzymes, and in fact some other proteins from the pancreas that aren't listed here. So these then were, if you will, providing a basis for the application of X-ray diffraction. If X-ray diffractionists could be found, in fact, that were interested in working on such a difficult project as a protein. 1935, we have the first crystals of a virus, and these were not really true crystals. These were what we might call paracrystalline materials. But nonetheless, they were, to most scientists, accepted as crystals. These were of tobacco mosaic virus, and they were simply obtained by centrifugation. In other words, Stanley simply put his extracts of the tobacco leaves, which were heavily laden with these virus particles, centrifuged down the virus particles, and by concentrating them to a high degree in the bottom of the centrifuge tube, he found that they would spontaneously crystallize out. So those were the first virus crystals, and in fact, those provided the samples for the very first viral X-ray crystallography. 1935 was also the year that Bernal, Crowfoot, and Fankuchen, you've heard those names already this morning, first began to apply X-ray diffraction analysis to protein crystals. And Dorothy Hodgkins, of course, looked first at pepsin and was able to show that when you dehydrate the pepsin crystals, when you took the water out or you let them dry out, then you lost the diffraction pattern. So she was the first to really figure out that you had to maintain them in a hydrated state during the X-ray diffraction experiment. Otherwise, you wouldn't get any diffraction at all. And then she obtained X-ray diffraction patterns of pepsin, which were quite remarkable in their time because of the very primitive kinds of X-ray spectrometers and X-ray diffraction devices that were available. And here is a photograph, for those of you who haven't seen this before. Here's a photograph of four people. There's Isidore Fankuchen on the left, on your left. There's Dorothy Hodgkins, there's J.D. Bernal, and there's Fankuchen's wife, Dina Fankuchen. And this is taken in the garden of Dorothy Hodgkins right here. And this was taken in 1939, just before Dorothy Hodgkins began her work on penicillin, just before J.D. Bernal went to advise Lord Mountbatten on how to manage the war effort. And Isidore and his wife returned to the United States very shortly after. He to Brooklyn Polytechnic, where he in fact inspired a whole generation of American macromolecular crystallographers. So those were some of the primal people in this entire field. 1937, I have to mention this, they crystallized lysozyme, which of course has become the model system for crystallization studies, or for at least a very long time has been the model system for that. They did this simply by taking egg whites and adding ammonium sulfate to the egg whites. So they were able to demonstrate not only that they could crystallize lysozyme, but they also showed that they could actually crystallize a protein from a very, very impure mixture, if it was the right protein. We all wish we had those kinds of proteins. Of course we don't. But they were able to do that. And once they had done that, of course, then they could purify further, and then they got these remarkable large crystals. In 1942, there was a paper that appeared, and again, when I was a postdoc at MIT and I was scrounging through the libraries for these old papers, I ran across this paper, long forgotten, but to me it marks a real turning point in macromolecular crystallization. Because for me it indicates that at long last, people began to understand the importance of growing protein crystals, and particularly large protein crystals. And Bailey, Kenneth Bailey, in 1942, and notice that this is at the height of the war, Kenneth Bailey had a paper, excuse me, in the Proceedings of the Royal Society entitled The Growth of Large Protein Crystals. And in that paper, he described how to grow large protein crystals of about half a dozen different protein molecules. And I think that's very important. Now, remember that during this period, we also had the development of an understanding of proteins themselves. We had Edsel, we had Ferry, we had Cohn, people like that who were studying the fundamental chemical structure, the fundamental chemical properties of proteins. And we were beginning to understand what their makeup was, what their functions were, what their physical properties, what their chemical properties were. And of course, one of the people who was instrumental in all of this was Linus Pauling, who also produced, through his students, a whole generation of X-ray crystallographers. This is an early picture of Linus Pauling right here. That is not a bottle of vitamin C that he's pointing at right there. It's a bottle of something else that I can't identify. But nonetheless, Linus Pauling was the first to really look at these proteins using the work of himself and Corey, his colleague Corey at Caltech, to begin to look at these diffraction patterns and to begin to look at the structures of the small peptides and to note the properties of the peptide bond, to note the properties of hydrogen bonding within these macromolecules. He was the one who proposed the existence of alpha helices in proteins. In other words, he was very, very instrumental in sort of setting the stage for our understanding of macromolecules and macromolecular crystallography. 1959, here again is where crystal growth merges with X-ray crystallography of macromolecules. 1959, with publications by Kendrew of the structure of myoglobin and by Max Perutz of the structure of hemoglobin, of course, hemoglobin. Those were the first X-ray structures of proteins. And those, of course, were dramatic events from the standpoint of molecular biology, from the standpoint of structural biology. The myoglobin was immortalized by this image drawn by Irving Geis of myoglobin. You see the heme group embedded in the center of that. And I think this was an important event because it brought forth the beauty, the inherent beauty of chemistry that was embedded or inherent in these macromolecules. We could look upon them almost as works of scientific art. Here's a picture of Kendrew, and he's building a Kendrew model. This is an experience people of my generation are familiar with. Fortunately for you younger people, this is no longer necessary. You have graphic systems. I always thought of this as something that if it didn't require two hands, you should have a glass of bourbon in one hand. Because it was an awfully tedious, incredibly demanding kind of thing to do. And that's Kendrew, who even when he was young, even when he was a young professor, still looked old and distinguished, as you see here. Max Perutz, who really is the father of X-ray macromolecular crystallography. Max, he was the mentor of an innumerable number of young crystallographers. He provided the sources for most of the crystallography laboratories in the world coming from his outfit. And he was a true brilliant scientist. I need not say anything more about him. I think everyone's familiar with that. Here's a remarkable photograph. In case you haven't seen this, is a remarkable photograph right here because it shows a collection of almost all Nobel Prize winners. And you may recognize here among them, here's Sir John Kendrew, here's Max Perutz, there's Francis Crick, here is Frederick Sanger, Hugh Huxley over here, and this character right here, this is Sidney Brenner, who one of the most brilliant geneticists of the 20th century, who was awarded the Nobel Prize just a few years ago. And you can see that from the joviality here of all of these people in this image that someone has just told a hilarious joke about tetragonal space groups. And I was looking for a smug smile on Fred Sanger's face, but I didn't see it, because Fred Sanger, of course, won 2 Nobel Prizes, so he had the right to say, well, I have 2, they only have 1. Hugh Huxley, unfortunately, never won that Nobel Prize. But this photograph was taken before Fred won his 2nd Nobel Prize for the sequencing of Phi-X174 DNA. Hugh Huxley is clearly the most dapper and urbane of the characters in this photograph. He's the only one who didn't get the Nobel Prize, and there may be a lesson there, I'm not sure. So what happened after this? Well, of course, a whole series of major events happened after that. The structure of insulin appeared, the structure of lysozyme appeared, structure of ribonuclease, trypsin, all of those molecules followed. Mostly those were crystals which were taking advantage of that work that had occurred back in the 1930s and the 1940s. That work of Sumner, that work of Northrop, and others of that period. So they were utilizing all of those crystals. They weren't generating new crystals themselves, but simply studying the crystals that existed. Now, an important thing happened in the 1969-1970 time frame there, and that was the crystallization of transfer RNA. And this was important for 2 reasons. First of all, it was important because this was the first RNA molecule to be crystallized. And what it demonstrated really was that RNA could be crystallized just as proteins, just using the same technology, the same techniques as proteins. And then, in fact, the crystals, when one looked at them with X-ray diffraction analysis, looked very much like, those diffraction patterns looked very much like protein diffraction patterns. So it was clear there was no reason we couldn't apply the same techniques, the same X-ray crystallographic techniques, to tRNA crystals and other RNA molecules that we had to proteins. The other thing that was important was that there was a very serious competition among groups at this time over the structure of tRNA. These competing groups were all trying to be the first to solve tRNA. And it's still controversial who was the first, but I happen to be part of one of the groups that were involved in this, so I don't want to give you my opinion. That would not be completely unbiased. But nonetheless, there wasn't this intense competition. And thinking back on it, what I would claim is that the competitors were the competitors simply because they had access to material one way or the other that no one else did. The group at MIT, for example, when Boehringer Mannheim first began selling yeast phenylalanine tRNA, we were able to purchase entire lots of this, which made it possible for us to now work with half-gram quantities. And back then, we worked with gram and half-gram quantities of these things. We were able to work with that large amount of material and to develop very good crystals. And similarly, Klug's group at the MRC were able to get large amounts of the tRNA, and they were able to develop good crystals of a different space group, different unit cell that they worked on. And Sunderlingham's people. And then there were Sigler's people in Chicago. They made their own tRNA. They made the FMET tRNA. But in a sense, the competition was restricted to those groups who knew how to get the material and grow the crystals. Not the best crystallographers by any means, but perhaps the people who knew how to get the goods. And that was what counted. And that's where I think a major philosophical change came into crystallography. That the idea was, how do you get the crystals, not necessarily how do you solve them after that, although that remained, of course, important. So it was a realization that macromolecular crystallization was important to structural biology, and this occurred between about 1970 and 1975. Now I'd like to describe, there's 2 what I would almost call beneficent acts of God. Some people would say, no, Mother Nature was being kind. Some would just say these were triumphs of technology. But there were 2 things that happened. These are collections of events, really, that happened. But these are 2 extraordinarily important things, both for crystallization of macromolecules, but also for X-ray crystallography and structural biology. And the first occurred in this period between 1975 and 1985. And what happened during that period was that almost independently, but almost simultaneously, synchrotrons were developed, which gave us high-intensity sources and tunable wavelengths. Area detectors were devised, which allowed us to collect massive amounts of data very, very rapidly. High-speed computers appeared, which allowed us to carry out crystallographic procedures at a very, very rapid pace there. And automated procedures came on board, and then finally cryocrystallography. And all of these things appeared almost in the same time frame. And if you look at this, one of these would not have been valuable without the other. Synchrotrons would have been pretty useless for the most part without area detectors or without cryocrystallography. And similarly, the high-speed computers would have been unusable if, in fact, the data hadn't been produced by the synchrotrons and the area detectors at such a rapid rate. So all these things were interdependent and yet developed, in a sense, in a completely independent manner, but all at the same time. So this is what I would call one of the first miracles here, is that all of these necessary technologies came together at exactly the right time in the 1980s. And here's a photograph. This is a remarkable photograph, really. It was taken on a synchrotron. It's remarkable because I actually made this photograph myself. So it is of a virus crystal. It's a virus crystal, and it just shows you, in a way, what you can do with today's modern detectors and today's modern synchrotrons. It's a virus crystal, and if you follow the pattern out, and you ask, and this is, in fact, we had to show 3 different exposures of the image in order to show the distribution of reflection, but if you follow the pattern out, it goes all the way out to 1.4 angstroms resolution. Now you think to yourself, a virus crystal, here we have 1.5 megadaltons of material that makes up the asymmetric unit, or makes up the unit of the crystal, and is producing X-ray diffraction patterns that we can record accurately, and then manage the 2.8 million reflections that were measured. We can manage that using those high-speed computers. So that's one of the first miracles that I would really say occurred in the second half of the 20th century. And the other one, of course, was the development of recombinant DNA technology. Because as I pointed out, all of the crystallography up until the 70s was being done on those proteins that had been isolated and crystallized back in the 1930s and the 1940s. Why were those proteins the ones that were isolated and crystallized? Because they occurred in very large abundance. If you look in the pancreas, you'll find grams of trypsin. If you look in the pancreas, you'll find grams of ribonuclease and so forth. So these were proteins that occurred in very, very high abundance. And that was true of almost all of the structures that were solved up until about 1980. But then, with the advent of recombinant DNA technology, we could take proteins which were barely detectable in cells, and if we could isolate their genes, which we could do, and clone them, then we could produce excuse me, in very, very large amounts, large enough amounts that we could now do X-ray crystallography. And this is what really opened up the gates of X-ray crystallography to really important biological problems. And as a consequence of that, you might say that DNA technology, or recombinant DNA technology, transformed macromolecular crystallography. But at the same time, I think it's fair to say that macromolecular crystallography has totally transformed the field of molecular biology at the same time. So those are my 2 acts of God, or my two technological triumphs that I think are absolutely important. 1985, we had the first, what we now call the ICCBM meetings. This was a meeting of macromolecular crystal growers. It was held at Stanford University. I helped organize this with another professor from Stanford University. And this is the first time that we actually started bringing together macromolecular crystallographers to talk about how to grow better crystals. And there's now something like 18 of these meetings that have been held subsequently every 2 years since that time. I might also point out that this is about the time, a couple of years later, that the Cold Spring Harbor course in macromolecular crystallography, or diffraction science, was begun by Jim Flugrath. Jim's here somewhere, but Jim really should get credit for this. But because of the Cold Spring Harbor course, now over 500 different students have been trained in how to grow protein crystals and how to analyze those crystals using X-ray diffraction analysis. And of course, there's other programs as well. Bob Sweet has a program now at Brookhaven on how to collect data from these crystals. There are workshops all over the world. There's one in Hamburg that's been run by Embl. It's now in its 18th manifestation. So there are many of these workshops and different kinds of schools that have developed over time, which have instructed people now how to go about this effort. And the next thing is something that some folks might not appreciate, but I do. And this was the introduction of the first commercially available protein crystallization screening kits. Now you might say, okay, so finally someone got around to selling these things that we needed. Well, it was much, much more than that. I once heard someone say that, you know, they should have given the Nobel Prize to the person who started the Sigma Biochemical Company, because Sigma Biochemicals has probably done more to promote biochemistry in the world than anything else that we can think of. And that's a fairly good argument. But I would make exactly the same argument for these commercial crystallization kits, because they in fact opened up X-ray crystallography. They opened up macromolecular crystallography to that great world out there who could only look on and wish that they could solve protein structures. But with the appearance of these kits that anyone could buy for a very low price, they could try to crystallize their macromolecule, and if they succeeded, then they could call themselves protein crystallographers. And I think this was the next thing that really opened up the field of X-ray crystallography, was the fact now the common man, the great rabble and riffraff who weren't trained biochemists, trained X-ray crystallographers, could in fact come in and participate in this great enterprise. In 1992, MAD phasing was introduced. Again, this was synchrotron dependent. You couldn't have had MAD phasing. Wayne Hendrickson, of course, mainly, and Martha Teeter were the first to show that anomalous dispersion was really a powerful technique for solving protein structures, and MAD phasing developed and SAD phasing after that. So that was another important thing that occurred, because it meant that you could solve the structure of a crystal without making necessarily a lot of heavy atom derivatives and not doing a lot of serious biochemistry. You could take, now using sulfur SAD, you can take the native protein in many cases and just simply use the anomalous dispersion from the sulfur atoms in order to solve structures. So these new phasing methods that appeared were also powerful promoters, I think. And then we had structural genomics and high throughput crystallography. So here's a timeline here. Here's a timeline from art to science, which more or less incorporates what I've already told you this morning. It shows you the various pathways here, and you see that it's drawn as sort of an exponential curve. Really, that should have a much greater slope than it does. I didn't know there was 100,000 now structures in the PDP, but in fact, this stopped in 2004. I think if we had continued it on beyond 2004, it would be somewhere up here above the screen at that time. And this reflects not only the expertise of X-ray crystallographers, not only their greater ability to solve structures, but it clearly implies a much, much greater ability to grow the protein crystals and to grow the macromolecular crystals that are necessary in order to supply or order to provide for those crystallographers. Now, in the last 25 years, there has been a turning, and it began really in 1985 with that, or 1985 with that first crystallization meeting at Stanford. But at that time, the people who were trying to grow protein crystals were doing more or less empirical kinds of things. And in 1985, some very, very important people like Franz Rosenberger and Alexander Chernov were brought into the field from the small molecule field, and they were determined to teach us protein chemists and us protein crystallographers how to really grow crystals, how to study the physics of the crystals, how to study them in a scientific manner, and to try to put protein crystallography and protein crystal growth on the same basis that small molecule or conventional molecule crystallography was on. And these people had an enormous influence because they inspired a lot of us to start using technologies that previously we'd never used in studying protein crystal growth. So the question is, well, what did we learn from all of this? And I once remember Brian Matthews said, well, you've learned a great deal, but was it really very useful? I mean, do we grow better crystals because of it? Or is it just new knowledge? Well, I can't make a strong argument. I can't point to certain enormous monumental triumphs that arose because of these specific studies. But I will point out that we're growing a lot more protein and macromolecular crystals now than we ever have before. And I have to ascribe at least some of that increased ability to do so to these studies and the information that we've gotten from these biophysical studies. So let me just quickly run through these. What have we learned from these different kinds of studies in the last 25 years? Well, from static and quasi-light scattering investigations, which were begun back in the 1980s, Bill Wilson, for example, at Mississippi State, has shown that the second virial coefficient of macromolecular mother liquors, this is something you can measure just with static light scattering, can be used as a predictor of success of specific crystallization conditions. And he defined what we now know as what he calls the crystallization slot. That if a mother liquor has particular properties that you can measure by scattering, then in fact you have a very good chance of getting a crystal from that. We've learned the kinetics for the formation of pre-nuclear clusters and their ripening into crystal nuclei. We now understand much better that process of nucleation. This again came from quasi-elastic light scattering. We know the sizes of critical nuclei as a function, for example, of the molecular weight or the diameter of protein molecules, so that we can estimate now what sizes these pre-nuclear aggregates must achieve. We have estimates, reasonably good estimates now, of the thermodynamic parameters that govern nucleation. These again from light scattering. And we have used light scattering to discriminate monodispersed from polydispersed macromolecular mother liquors. And this is important because we now know that this monodispersity is an important condition if you're going to grow macromolecular crystals. It's extremely difficult to grow them if you have a polydispersed solution. And we can use light scattering, in fact, to detect that. From calorimetry and thermophore experiments, and some of you are using thermophores in your own laboratories, we can identify factors such as pH, buffer, certain ions, certain small molecules that stabilize the proteins in the mother liquors, and predict for us that those might be useful things to include when we try to crystallize. We have found the correlation, if you will, between protein stability and crystallization. We've demonstrated that, and we've got measures now of the enthalpic and entropic contributions to the formation of protein crystals, and something about the contribution of solvent. And Professor Derwende can tell you much about, I think, that. He has the opportunity. If we get you into a corner, he'll tell you much more than you want to know about it, but he has certainly shown that it is important. What have we gotten from atomic force microscopy? Well, I won't go over all of these things because you can read for yourselves, and there's going to be a paper, it was a transaction paper, and I've included these here, and you can read them for yourselves there. What have we gotten from all of this? Well, now we know what the mechanisms of macromolecular protein crystal growth are, or macromolecular crystal growth are, and I include here viruses and nucleic acids, of course, as well as proteins. So we know the mechanisms. We know about screw dislocations, two-dimensional nucleation. We know about normal growth. We know about all of these. We know a lot about the defects that are formed in the macromolecular crystals. We know about the stacking faults and the vacancies and the different kinds of line dislocations that we find in them. We have a much better appreciation of the kinds, the variety, and the damage that is done by the incorporation of impurities into macromolecular crystals. All of these things we can actually visualize and see using atomic force microscopy. We've gotten, again, an additional set of thermodynamic and kinetic variables from these kinds of studies. We now know why the protein crystals stop growing, what the source of the growth termination is due to the formation of these surface impurity layers, and we've shown, in fact, that you can incorporate small crystals into large crystals, something which is remarkable in itself. What do we get from gel and microgravity studies? Some of you would say, well, we get nothing from microgravity studies. Others of you, like myself, would argue that we've gotten actually quite a lot from microgravity studies, from doing things in space. And in particular, what we've gotten is we've gotten estimates or measures of the relative roles of convection and diffusion in the transport of molecules, macromolecules, to the surfaces of growing crystals. And you might say, well, who cares how they get there as long as they get into the crystal? But the truth of the matter is how the molecules reach the crystal surface and how they are incorporated in that process is very important to the perfection of the crystals, the sizes of the crystals, and the general properties of those. So we now know the effects, if we will, of gravity on crystal growth on the Earth. We have genetic experiments. I said these were biochemical, but in fact, we have a lot of mutation and truncation experiments. We now know how to couple mass spectrometry to genetics in such a way that we can predict domains, and we can make truncations, or we can make genetic constructs. We know that we can use proteolysis in many cases in order to get protein crystals that otherwise would not crystallize. Todd Yates and others have shown that the importance of molecular symmetry in crystallization. They've made genetic constructs which in fact have higher symmetry than the native molecule, and they demonstrate in fact that that higher symmetry leads to enhanced crystallization, and that's important. And then the use of antibody fragments and ancillary molecules which can be coupled to intractable cases, like membrane proteins in many instances, and these can help, if you will, lead the target molecule into the crystalline state. And then the final slide I'm going to show you right here is from investigations of the chemistry of crystallization. Well, we have new precipitating agents. We have a lot of precipitating agents now that we weren't using 25 or 30 years ago. We have polyethylene glycol and peg methyl monoester and a whole variety of different pegs of different sorts. We have jeffamine. We use malonate now. Taxamates come out, which is a variety of different small organic acids. We have methylpentane diol, thanks to Murray Vernon King's work. We have a lot of new precipitating agents and crystallization agents that we didn't have before. We have a lot of new additives and conventional small molecules. Bob Cudney and myself worked on this very hard for a number of years. We call them silver bullets, and these are additives which enhance the formation of the crystal lattice. And these, of course, were very important, I think, in a number of cases in getting protein crystals. We've shown that the surfaces on which crystals grow are important, that there is epitaxy in some cases, or there is promotion, heteroepitaxy in others, where a surface promotes the formation or the nucleation of crystals. And this is clearly important because it's been reported, for example, that many macromolecular crystals will only grow on epoxy resins, or on human hair, or on rabbit hair, or they will only grow on etched graphite, and so forth. So it is important that these surfaces be considered when you're crystallizing macromolecules. The demonstration that specific buffers and specific ions influence the crystallization process. I think Janet Newman gets a lot of credit for this because she was the first one to argue that, yes, you've got pH 7.3, but what buffer did you use to get pH 7.3? And I always would have said, well, it really doesn't matter. One buffer is another buffer. But in fact, that's not the case. There are buffer-specific proteins where one buffer will work and the same buffer, another buffer at the same pH fails. We have a lot of new detergents and ampliphiles which have become available to us. I think this has almost become a problem. We have so many new detergents and ampliphiles, we don't know which ones to use in most cases, but nonetheless, the availability of these is important. We have the cubic lipidic phase. It's been developed for the crystallization of membrane proteins, and that's been extremely successful, particularly in recent years. I think that has really shown itself to be a valuable tool. And finally, I think from all of these studies, from the collection of all of these studies, we've come to the realization that the protein that we're trying to crystallize is itself the most important variable in the entire process. That in fact we can gain far more by paying attention to the macromolecule we're working on. Its biochemistry, its stability, its properties, its genetic constructs, its truncations, and so forth. That really provides us with the best set of variables and the most powerful variables for, in fact, enhancing crystal growth. And I think I've used up all my time. Yes. Thank you very much for your attention and coming this morning.
We started the session a couple of minutes late, so I think we've actually got time for possibly a couple of quick questions if anyone would like to ask anything.
I've often thought that it would be a dream result if we could take the amino acid sequence of a protein and predict the best crystallization protocol. Do we have any progress toward this dream?
Not much, but I will say one thing we can do from amino acid sequences, a couple of things we can do. We can look at an amino acid sequence, and now, with some confidence, we can find unstructured regions. And we can predict what are unstructured regions. And if we can remove those unstructured regions or somehow minimize their influence, I think we can enhance the protein crystallization there. That's one thing we can do. Also, we can identify, just by looking at the sequence in general, what the surface residues are going to be. We know that the lysines and the glutamic acids and and the arginines are going to be on the surfaces. And as Professor DeWenda has pointed out, we can take advantage of those by in fact replacing those with other amino acids which lead to a less entropic loss when they are crystallized, and we can utilize that by using those mutations there. But in terms of actually predicting, I mean, the amino acid sequence, normally you can identify domains, and that will tell you where you might want to truncate. But in general, looking at the amino acid sequence and predicting, we've been trying to do that. I agree, we've been trying to do that forever. And I think that's about the only progress I can report at this time.
Just a few words. So there is some progress in terms of being able to assess in silico the crystallizability based on all these parameters. And so we're working with Adam Gajic to get the XTAL-PRED algorithm being more reliable. And in fact, those predictions regarding crystallizability, when you take all these physical parameters, it's getting better. The correlation between crystallizability and the right conditions is the most difficult question here.
Yeah. And you know, we had high hopes that when we had a data bank large enough, we'd be able to look at that data bank and certain things would become obvious. Like if we had certain amino acid sequences or certain proteins or certain this or certain that, well, we would know how to crystallize those. Well, I see someone roaring to the front here to make some comments.
I want to report that we actually have a paper submitted with Lukas Kruger lab. We have analyzed over 8 million protein sequences in nearly 2,000 genomes. And in every family you can find a protein that has higher crystallization propensity.
Yeah.
So I think this is, as we accumulate data, I think we'll be able to predict much better.
Yeah.
Which including GPCR.
I think there's no question that stability is correlated with crystallizability. So if you can somehow deduce from the amino acid sequence, maybe from thermophore experiments is even more direct, but if you can deduce the stability of the protein, that gives you one mark. The other thing is this idea of symmetry. There's no question that symmetry helps. No question. And that's why icosahedral viruses are wonderful to work on. I started working on icosahedral viruses 20 years ago when I realized this. They crystallize so easily, for the most part, because of that very high symmetry. And I think that's true in many of these macromolecular complexes. If you can find some symmetry there to take advantage of, that's important as well. So stability and symmetry, those are some important areas. But exactly how to predict the conditions, that I can't say we've really made a lot of progress.
Okay, well, thank you very much, Alex.
Thank you.